So just a brief introduction about myself, as I don't have any slide for that. It's my great pleasure to be here today, and thank you for the invitation from STRAP. I'm a senior lecturer. I'm by background computer scientist. So I'm a computer scientist who speaks biology and biomedicine. So I have background in machine learning, artificial intelligence, and has been data that I'm working on biological. These are joined by waves of information from other omics data, clinical trials, and patient records. Analysis of this big data is launching the era of precision medicine. I read that slowly because I would like to explain every bit of this sentence. So these big data efforts face huge challenges, of course, from creating analytical tools and solving scientific puzzles to accessing millions of gigabytes of data and overcoming barriers to access patients' records. There's a lot of ethics and privacy barriers here as well. So we have a very general audience today, and I would like to go through important bits of this sentence. What do I mean by sequencing technologies? What are genomics overall? What are other omics data, and how they lead to precision medicine? So what is genomic? Your body has, your body has close to 37 trillions of cells. Within your cell, you have nucleus, and within nucleus, you have chromosomes. Each chromosome is made up of a protein and a single long-strand molecule of deoxyribonucleic acid called DNA. DNA is composed of 4 nucleotide bases, which is abbreviated as A for adenine, T as thymine, C as cytosine, and G as guanine. And a genome consists of all of the DNA contained in your cell's nucleus. So basically your genome is like a blueprint, containing all of the instruction needed to make and maintain the function of every cell, tissue, and organ in your body. And in the human genome is basically made up of over 3 billion of such base pairs. So what is genome sequencing? Just simply, in simple words, the genome sequencing is to reading out and figuring out the order of DNA nucleotides or bases on the genome, which we basically would like to see that what are the orders of A, C's, G's, and T's. DNA sequencing has done by very high-tech machines called high-performance technologies or next-generation sequencing technologies that I'm going to briefly talk about it, which basically reads a sequence of DNA bases. And these sequences of DNA bases are translated from live chemical alphabets, which are that 4 different nucleotides that I just described, to alphabets that is written in English. So, so you can see like this. This is a portion of a DNA, which has adenine followed by guanine, and then thymine, and then cytosine, and it follows up. Genome sequencing is often compared by decoding. So we would like to decode our genome and see what are the letters within that. But it's basically quite in code yet. It is like a mysterious language. Let's have a quick detour, and a fun detour, and a bit of brain exercising for all of you. Imagine that genome is a book written without capitalization, punctuations, without breaks between words, sentences, paragraphs, and with strings of nonsense letters scattered between and even within words and sentences. A passage from such book in English can look like this. I give you a few seconds to have a read through. Can you get any information out of that? Can you read any words? What? Quick. Good. And this is the passage. A quick brown fox jumped over the lazy duck. The duck lay quietly dreaming of dinner. And this is what is hidden within this passage. And this is pretty much like what is hidden in DNA. So sequencing the genome doesn't immediately lay open the genetic secrets of an entire species. Even with the complete human genome in hand that we have currently, scientists still have to translate those strings of letters into understanding of how genome is working. what are various genes that made up of genome, what they are doing, what are differences between different genomes, and how these differences are translated to the phenotype, to the differences between us. So that is just a primer, a very quick primer in genome sequencing, a bit like more technical than passage of English books. So the genome are very large, and at least with the technologies that we have currently, we cannot read it out from the end to end. So rather, what a current technology is doing is going to chunk that to some pieces, initially very small pieces, now with some new technologies into larger pieces. But then again, with some chemical reactions, we can identify the order of these pieces, and the outcome of that would be sequence reads that you'll see here. Then sequence reads by computationally will be aligned to a reference sequence and will give you eventually your whole genome. The cost of sequencing a human genome has been substantially dropped. The first human genome sequence as part of Human Genome Project in late 19th century. The project ended up taking 13 years with a cost of 2.7 billion. That was huge, but that has been dropped substantially. So now in 2020, we can have a whole genome sequence in an order of minutes with a cost of less than 1,000. And that basically simply says us that once we have reduced cost, there will be growth of DNA sequencing. So, so there is some sort of thing that is under that, but that's fine. The capacity with the reduced cost and our capacity to increase the generation of sequencing data has been substantial. Oops, I just want to, oops, sorry. Yeah, how can I have it?
Orange one, height. Height floating. Down.
I see. Okay, good. Thank you. So there is roughly 6 orders of magnitude in the past 7 or 8 years of our ability to generate data. So you'll see here a plot up to about 2016 with milestones of what has been done. So that was the first like Sanger project here. Then the first human genome has been sequenced in about 2009. And then there was 1,000 genome, and these are milestone TCGA, the Cancer Genome Atlas, EXAC, which is Exome Association Consortium, and then it continues. This study has been up till like 2016 and has been projected the growth of the data up until 2025. And you'll see here, this is the amount of data worldwide, the annual sequencing capacity is per year. And you'll see that we are in the order of zettabyte here. So I would like to compare genomic data with other domains of big data, which basically 3 main domains that I'll discuss here. So we have astronomy, which is just covered with Catherine. We have Twitter and YouTube, 3 main domains of big data. Twitter has come to scene in 2006, and you all know that it's generating astonishing amount of text data. And YouTube has come to scene in 2005. And now it has been generated extraordinary interest worldwide to generating huge number of videos basically and medias. How it compares with the genomic. So astronomy is the oldest one. It's like 20 years now that astronomy is in the field of big data. All the other fields are just emerging, including genomics. Is generating, and these are like estimations for 2025 based on the projections, the historical projections. There are 25 zettabytes per year of data generation in astronomy, and that ended up to 1 exabyte per year of data storage. And you'll see here that, as discussed by Katharina, there are like some sort of in situ data reduction, that the amount of storage is less than what has been generated as estimated by this study. Twitter, the estimation is to generate up to 15 billion of tweets per year. That is equivalent to up to 27 petabytes per year of text data. and U2, 0.9 billion hours per year, equivalent to 2 exabytes per year. And on top of all is genomics, 1 zettabases, which is 10 to the power of 21 per year, which can be up to 40 exabytes per year. But this is just the beginning of the story. And genomic is just one single part of the data. We need to move from genome to proteome and up to phenome. This figure will basically show the central dogma of molecular biology, the flow of genetic information from DNA to RNA and then to functional products, which is usually protein. So here we have DNA. Everything that we talked about sequencing genome was about sequencing DNA, ACTGs. The next step, this DNA would be transcribed to RNAs, and these RNAs would be translated to proteins. And we need to capture the information along the way. And this is not just these 3 steps. There are multiple intermediate steps as well. And this is sequencing beyond DNA genome. Various sequencing methods can quantify several steps of chromatin modification and also like changes in DNA between the cascade of information from transcription to translation and providing an integrated personal omics profile to map all regulatory and functional elements of an individual, which basically leads to personalized and predictive medicine that I'm going to discuss in future slides very briefly. But that gives us basically multi-omics data. Genomics is the very first level. We have epigenomics on top of that with other sequencing technologies. We have transcriptomics, and this transcriptomics contains lots of different transcripts from those that encoded to genes, encoding genes, and those that they are not. Then with different technologies, mostly mass spectrometry technologies, we have proteomics, metabolomics, lipidomics, glycomics, phosphoproteomics, and that goes on until we reach to phenome and the environment. This is multi-omics data generation, and you can imagine how much amount of data would be generated across different levels. On top of that are electronic health recorded, real-time patient health data, and all the variables that now has been generating lots of data for every single individuals. So all this data are coming together, and precision medicine is foreseen to put together all this data for us to have a better preventive, predictive, and diagnostic medicine. So this is a very simple workflow of how precision medicine works. We generate multi-omics data from a population, but instead of the common practice that has been there for years, that we have a population and we prescribe some sort of treatments for the population, it will work for majority of them, let's say about 50%, but for many of them it fails because it is just goes with the rule of the average of the population. Precision medicine and personalized medicine would like to look into each individual, look into genomics, transcriptomics, epigenomics, and beyond of each individual to be able to trigger treatment, and not just treatment, before treatment, to be able to diagnose disease, to be able to have early diagnosis that I'm going to discuss briefly on that, and eventually prevent, because if we diagnose quite early, we can intervene effectively and we can prevent basically the disease to happen in a phenotypic level. So this data would be generated, and then here is when the data science will come and seen, and we would like to get some sort of interpretation and some sort of information out of this data, some sort of markers, we call it, that will give us, there are some sort of indicator of the state of the disease or the possibility of the patient to respond to a treatment or not responding to a specific treatment. These type of signatures or biomarkers will help us to certify patients to different groups and make the treatment specialized for that specific person. And this is what we call it precision or personalized medicine. Sorry. So biomarker discovery is integral for precision medicine. As I just explained, we need to identify markers. Biomarkers basically stands for biological markers or biomolecular markers that can tell us that what is disease and what is health. for every single individual. Let's say it's just a very like cartonic view. Of course, it's much more complicated than this. But this set of patients, these have like A and B, assume that there are some mutations or some specific molecules in the derived from blood or other tissues of the patients. A and B are high, but C is low, while here we have A and B low, while C is high. And then that would be two subclasses that the treatment would be triggered according to that. So biomarker discovery, lots of molecules, a lot of information, how we can grasp information out of that. And this is, well, you know, like teams like me within my university and across the global common scene to grasp information out of this data. What are the molecules that they can predict the outcome for us? So very briefly about biomarker discovery. One of the frontiers in the field of biomarker discovery is about identifying these biomarkers from some fluids, and specifically blood from body, which we call it liquid biopsy. So the tradition previously has been tissue biopsy. Let's assume for cancer, which tissue biopsy is quite like prevalent. The standard is to get some bits of the tumor and then analyze that bit about the molecular composition. This is very invasive, this is not repeatable, and this comes with a lot of side effects. And imagine for some cancer types, let's say glioblastoma, we need either to cut a scalp or have some sort of drill within a scalp to get some piece of tumor within brain. That's not something repeatable. And at the same time, even once we get it, we can't capture heterogeneity because we just got one specific site of that tumor. Well, tumor, everyone knows that it's quite heterogeneous. So the future of like precision medicine moving forward from tissue biopsy to liquid biopsy, and in particular blood biopsy, which is a simple blood test which can be incorporated into any routine, any like medical routine. You'll go with blood test per year, and you can have like consider that as liquid biopsy to be tested for lots of different diseases, some sort of nasty diseases. Tissue biopsy is invasive, liquid biopsy is minimally invasive or non-invasive. It comes with pain, it has minimal pain or risk. It's not easily obtained, it's easy to obtain, it's difficult to repeat. The other one is repeatable, failure to reflect heterogeneity, while liquid biopsy represent heterogeneity because it comes from blood from the whole tumor. And it fails to detect metastasis, well, that one gives us the comprehensive sort of view. But the problem is that identifying biomarkers from blood is finding a needle within a haystack. The yield is low and the signal is very low, while it's subtle, and it's quite hard to identify robust biomarkers from blood. And this is where we would like to incorporate latest advances in artificial intelligence, which can capture subtle changes between different subgroups to be able to identify these biomarkers in a robust and reproducible way. Just some examples of molecules within blood that can be used for biomarker and how it can be integratable. And some of like our recent studies, like last couple of years, on biomarker discovery and different types of cancers, as well as other diseases like multiple sclerosis. What are the challenges that exist? How much time do I have? Not much. Couple of minutes. So I move that forward. There are lots of like interesting things to discuss now, that what are the challenges and how we address that, but I'm going to skip that quickly. Specificity, the combinatorial exposure, like what are the different combinations that can be considered as biomarker, unused big data. That is a sort of major problem in genomics. Well, I think it's not in astronomy. Everyone around the world is generating data. There is a huge amount of data, but they are not coordinated and they are not reusable by others. We have lots of data, but are always nagging with lack of data because everyone would like to look into a specific condition. For instance, I would like to look into glioblastoma in response to specific treatment. There is no other data for that. And I can barely collect and sequence, let's say 30 samples. What can I do with 30 samples? Nothing. But when I go out there, there is a lot of data, how I can use this unused data for the purpose of improving our prediction. This is one of the challenges that we are working on very relentlessly. And multi-omics biomarker, we are generating lots of data across different levels, but our ability to integrate them is lacking, far behind, and require concentrated effort for us to be able to use this huge amount of data. Last topic to be discussed on single cell omics analysis, and it's quite related to big omics because that is another level. We talked about genomics, how big it is. We talked about multi-omics, and I would like to go one step further about single cell omics analysis. Let's compare bulk with single cell sequencing. It can be compared like a mix of berries and a smoothie. What happens in single cell sequencing, the previous practice, which we call it bulk sequencing? We get a sample, we get part of tissue of, like, let's say, tumor. We smash it together and get all the RNAs within that and try to sequence it. What is happening is what you see in that diagram below. We have lots of cells, but we get the average cell. It's neither blue nor red, it's just purple. It's the average representation. But within the last few years, there has been huge sort of improvement in moving on from bulk sequencing to single sequencing. I would like to show here as well that how important it is to have single sequencing. Let's assume this is a tumor, and we have different subclonal parts of tumor. They are different cell types. If we go with bulk sequencing, we get the average colon. The average colon is represented by red. We identify biomarker and the treatment, and we resolve that. But the cancer is still there, and that is why we see a lot of recurrence after treatment. If we go to single cell analysis, we can identify different composition and subclonal. We can identify subclonal specific biomarkers and their corresponding treatments, and the cancer would be completely removed. And this is how important it is to move on from bulk sequencing to single sequencing. Getting back again to 4 domains of big data, I would say that single cell omics generate ultra big data. A typical single cell RNA sequencing experiment will measure onto order of 20,000 transcripts on thousands, if not millions of cells. This is one single experiment. And you can imagine how it would be extrapolated to all the samples, all the patients, etc., once we have this technology settled down. And more importantly and more interestingly, we are moving now very rapidly from single cell sequencing to multi-omic single cell sequencing. From one single cell, current technologies can help us to understand the information from genome, methylome, chromatin features, transcriptomic, and proteomic all together. And you can imagine how much data it will generate. Just briefly about what we are doing in that sense. In some of the algorithm and some sort of everything crashes when we deal with this big data on just single data set of mouse of 1.3 million cells, single sample. All the method that we're applying for dimensionality reduction has crashes, except for some of the very linear, simple ones. And it requires a lot of effort, not just to improve our capacity to analyze this data in terms of computational resources, but also to ability to have scalable and fast methods that can be handled, can handle such big data. And that's it. This is like just research landscape in my lab, which I'm going to skip. And any question, I'm very happy to answer.
Thank you very much.
You're welcome.
Now, question, do we have questions online?
There is a question by Katrina Brand. What are some of the ethical challenges with this type of big data?
Yeah, that's a very good question.
Do you mind open here?
Yeah, the ethical challenges is mostly about the patient's information, because these information are revealing the identity of patients. And one of the major challenges are privacy issues. That, because once we have people's genome, we would like to do it for good, but we can do it for bad as well. And there is lots of like scary stories that come out of it. one of the major things that people do is the identification, of course, before using that, but it's not the whole thing. And there is a lot of, it's not my field that what are the ways to sort of address these privacy issues, but this is one of the obstacle that has been stymied the generation of the data, because we should resolve that in a way that has been standardized across the globe for us to make sure that these data has been used for good only.